first here. This one is showing you meiosis, okay? And in fact, it's showing you meiosis 1 and then meiosis 2. So let's go over meiosis 1 first. Now, the steps in meiosis 1 here, and by the way, it's not specifying male or female, it's just general meiotic, the meiotic divisions. Here you have prophase 1, early prophase 1, and you can see how the chromosomes have paired up, okay? Have synapsed. So this is late prophase 1. So all of this is prophase 1. And then finally, they will line up on the spindles here, the meiotic spindles, during this phase called metaphase 1. Now for metaphase 1, the next step is anaphase 1, and you can see how the chromosomes are separating towards opposite sides of the divided cell. And at the end of prophase, anaphase 1, you go into the last step, which is the completion of telophase 1. So this is actually the completion of telophase 1, produces 2 daughter cells that are now haploid. But these 2 daughter cells now will undergo the second meiotic division. So it is debatable whether we can say this is end of telophase 1 or beginning of prophase 2, because it looks like a type of prophase, type of a step. So you can say that this is prophase 2 as well. You can see they're in prophase 2. The cells now undergo meiosis 2, prophase 2, metaphase 2. See how the chromosomes lined up on the spindles? Then the chromosomes separate. This is anaphase 2. And then finally, at the end of anaphase 2, you have telophase 2. Okay? So here at the very end, you produced 4 daughter cells from one initial primary cell. Okay, the primary cell, which is either primary spermatocyte or primary oocyte. Okay, which resulted from mitosis, the mitosis of this oogonia or spermatogonia. Okay, so once again, prophase 1, metaphase 1, anaphase 1, completion of telophase 1, beginning of prophase 2, metaphase 2, anaphase 2, end of telophase 2, and you have here now your haploid cells called gametes. Okay? Now, let's look at this model. Maybe you want to pause for a second. Let's get this one done. So that model, keep in mind, is showing you specifically the phases of meiosis 1 and 2. Now here we're going to skip a lot of the phases. It's going to jump phases. It's not going to go directly every single phase, because then you would have to have a very big model. So what we'll start here is we'll start with those primary cells. So now you can start filling. We'll start here with the primary cells. Now this is the column for the male and for the female. On the male column here, we are actually observing spermatogenesis, the production of the sperm cells. Okay. This up here is our primary spermatocyte. The primary spermatocyte. Okay. The primary spermatocyte then, through meiosis 1, begins the different steps of meiosis 1. This looks like it is metaphase 1, anaphase 1, and then comes telophase 1, and at the end of telophase 1, what did you produce? You produced 2 new cells. These are your secondary spermatocytes. This is one secondary spermatocyte, already initiated meiosis 2. This is the other secondary spermatocyte, more advanced, because it's already in anaphase 2. So this one is just in metaphase 2. This one is more advanced, already in anaphase 2. But these are both haploid cells, the secondary spermatocytes. When they are finished completely dividing through meiosis 2, you will then have 4 daughter cells in total. 2 came from this spermatocyte, 2 came from this secondary spermatocyte, and these cells are now called spermatids. Now the spermatids are not finished yet, they will mature in a process called spermiogenesis, from this into this. These are your spermatozoa. So, your spermatids mature into spermatozoa through a process known as spermiogenesis right here. However, the whole process of producing the sperm cells, all of this process is spermatogenesis. Now, in this column you see oogenesis, the production of the egg cells. We'll start up here with the primary oocyte. The primary oocyte begins meiosis 1, okay, through the different phases, okay. This here is a primary oocyte in metaphase 1, meaning this has got to be after puberty. This is when puberty has already begun. Because remember, we said the cell will be arrested in prophase 1, before puberty. But this is already more advanced. This is metaphase 1 of the primary oocyte, and anaphase 1. And at the completion of meiosis 1, you will get a secondary oocyte, which is better represented here. Okay, see the secondary oocyte is forming, and a polar body is forming as well. So here you go, your secondary oocyte at the completion of meiosis 1, a secondary oocyte, and you have one polar body. Now the polar body immediately begins its meiosis. See the polar body? It's divided into 2 more polar bodies, and the secondary oocyte also will divide when fertilized. So when it's fertilized, the secondary oocyte will become an ovum and one extra polar body, which is forming, you can see right here, in this process. This looks like it is anaphase 2. Anaphase 2. Okay? So at the end you will have 1, 2, 3 polar bodies and one egg cell that becomes the ovum. This occurs if the egg cell is fertilized. Now the ovum is not yet complete. When it's fertilized, the egg cell becomes an ovum, and the ovum shortly afterwards will become the first new cell of every individual, called the zygote. So this here is the zygote. It's a diploid cell now. Okay, so here it's still haploid, and now here it's diploid, known as the zygote. Okay, I know this part is a little bit confusing. The model doesn't do it perfectly. However, one more time, this side's a little bit difficult. It's a primary oocyte in metaphase 1, primary oocyte in anaphase 1, secondary oocyte produced, secondary oocyte is fertilized, and it's forming now another polar body and 2 other polar bodies from the first original polar body. And then this secondary oocyte becomes the ovum. Okay? This again happens if there was fertilization occurred. So we are assuming that fertilization occurred here in this model. Finally, this becomes the zygote. Questions?